So, as you're doing from your biochemistry book, one correction that I have to make is in the previous pages, that is your page number 326 on my book, you have this diagram which is actually wrong. So, the correct diagram for this is this, the purine ring and its component. Firstly, we have the numbering from 1, 2, 3, 4, 5, 6. So, you number it from 1, 2, 3, 4, 5, and 6. Then you go for 7, 8, and 9. The carbon number, so aspartate is giving 1. Aspartate will give the nitrogen number 1. Then this 3 and 9, both of them will come from glutamine. 3 and 9 from glutamine. Then the carbon number 6 from carbon dioxide. 2 and 8 are coming from the N-formyl tetrahydrofolate, which are this is 2 and this is number 8. Sorry, this is number 8, 2 and 8. So you can see 3 and 9 from glutamine, 2 and 8 from NFTF, 1 from aspartate, 6 from carbon dioxide, and the rest from glycine. So this is the simple division that you need to know for the purine ring and its sources, which are 3 amino acids, 1 carbon dioxide, and N-formyl tetrahydrofolate. Now what happens is, after passing through the whole cycle, we get inosine monophosphate. This inosine monophosphate, also they are saying that they are giving 4 steps in this pathway require ATP. The main thing that you need to know about this is the total 5 ATPs are used in purine synthesis. If you want me to teach the whole cycle about this, like the whole inosine monophosphate pathway, you can let me know in the comments below. Firstly, I will go to this site that you know, from all the stuff that we made above, we get the inosine monophosphate. Now this inosine monophosphate will get converted into AMP and GMP. Let's see. Here they have written synthetic inhibitors, and the synthetic inhibitor is the sulfonamide, which actually inhibit the purine synthesis in the bacteria mainly, and the methotrexate, which inhibits the conversion of the dihydrofolate into tetrahydrofolate. I have discussed both of these in my previous video. Main thing that you need to know is that inhibitor of the purine synthesis are toxic to the tissues, because what they are going to do, purine synthesis is not taking place like in the humans for that, the folic acid, etc., but they are toxic to the tissues and the skin, the GI tract, the immune system, the hair follicles. As a result, individuals that are taking anti-cancer drugs can experience adverse effect, like they have GI tract disturbances. Immunodeficiency is the main thing, because the tetrahydrofolate is not formed. So the persons that are suffering from cancer, and if you give them anti-cancer drugs, firstly they are going to have their cell dividing inhibited in the most you can say painful manner. So we should always sympathize with the patients who are going through cancer, right? Now this figure number 22.8, it might seem very much dangerous to you, but it's not. Let me explain that to you in like 2 minutes. So first is inosine monophosphate. That was the main reaction that was formed by previously. So if we have to convert IMP into AMP, what we are going to do is that we'll form a middle compound, that is adenylosuccinate. And whenever a compound is formed, it is made by its own synthetase. So adenylosuccinate synthetase is the enzyme which will convert the IMP into adenylosuccinate. And the main things that are used in this are aspartate and GTP. Aspartate will be consumed, whereas GTP will be converted into GDP, and inorganic phosphate. So inosine monophosphate with the adenylosuccinate synthetase enzyme gets converted into adenylosuccinate using aspartate and GTP. Now we are going to remove fumarate from this adenylosuccinate, and this enzyme that is going to remove something will be the adenylosuccinate lyase enzyme. So adenylosuccinate lyase removes fumarate and we get the compound AMP. So IMP to AMP involves 2 steps. One is adenylosuccinate synthetase enzyme, other is adenylosuccinate lyase enzyme. With the synthetase we get the compound, with the lyase we remove fumarate from this compound to form AMP. Second thing, if we want to make GMP from it, in the previous step what we did was we used aspartate and GTP. What we are going to use in this, since it's a guanosine, so glutamine will be used. And the second thing is ATP will be used. So guanosine will have glutamine plus ATP, whereas the other one has aspartate plus GTP. Okay, A and G are both used. You can see A and G. So now inosine monophosphate gets converted into GMP by IMP dehydrogenase. Since it's a dehydrogenase enzyme, it's going to remove the hydrogen compound, hydrogen from this. So NAD comes, it takes 2 hydrogen from this compound, it gets converted into NAD, H plus H. So it gets 2 hydrogen compound removed from this. So it's a dehydrogenase enzyme confirming or transforming IMP into xanthosine monophosphate. Now this xanthosine monophosphate by addition of glutamine and ATP with the GMP synthetase enzyme gets converted into GMP. Simple as that. In this reaction, we firstly had synthetase. In this reaction, we had synthetase in the second step. So these are the two enzymes which are mainly used. So inosine monophosphate gets converted into first xanthosine monophosphate by the enzyme, the IMP dehydrogenase. Then this GMP synthetase converts it into GMP. Since it's a dehydrogenase, always remember NAD plus will be used, and xanthosine monophosphate gets converted into GMP synthetase. So for the G thing, we have glutamine, and for the synthetase, we have the ATP always. So guanosine monophosphate has been produced. One major thing that we have to discuss over there is the mycophenolic acid. Mycophenolic acid, which is actually an inhibitor of the IMP dehydrogenase. So what will this inhibitor do? It will prevent the synthesis of this GMP. So GMP synthesis will be prevented. T and B cells, which are the key components of the nucleic acid, they will be prevented, and mycophenolic acid will act as an immunosuppressant drug. So main thing was, in the previous thing we studied that if we inhibit the enzyme, the GAR formyl transferase, if we inhibit this enzyme and the dehydrofolate reductase enzyme, we can prevent cancer. Whereas if we prevent the IMP dehydrogenase enzyme with the mycophenolic acid, we can do an immunosuppressant, or we can prevent graft rejection. Very exciting and very much informative for me, and that is how I remembered all of this stuff. So let's meet in another video where we will discuss the salvage pathway. I hope you understood this IMP GMP pathway.